Introduction, Jason. As you said, I'm a master's student at the University of Regina, and I am doing my master's actually between two faculties, you'll see here, between kinesiology and health studies and the Faculty of Science. So it's because my research itself is kind of interdisciplinary, and that's what I'm going to talk about today. This is a project that I started working on during my honors, and I've continued working on it now for my master's. So this project centers around the gut microbiota and how it changes in age and how it's affected by a variety of lifestyle factors. So I'll get right into it. I'll start off with an introduction of the gut bacteria. So the gut microbiota can be considered the most essentially metabolically complex organ in the human body because it's comprised of the eukarya, bacteria, archaea, and viruses that live in our gut. My project specifically focuses on the bacteria because those are the ones that are the most prevalent and they have the biggest impact on human health. You can see here those dark blue boxes in the leftmost. These are the most dominant phyla in of our gut bacteria, and then those two in the box, Firmicutes and Bacteroidetes, these are the most abundant, comprising of approximately 90% of all the bacteria in our gut. So these gut bacteria play a significant role in human health as they produce numerous metabolites involved in regulating key functions of the body, such as nutrient processing, homeostasis, and even immunity. These bacterial genera circled here in purple are some of the ones that are associated with human health and proper gut functioning. So, key metabolites produced by the gut bacteria are short-chain fatty acids, or SCFAs, and the primary ones are shown here in the top, propionate, butyrate, and acetate, and they play significant roles in cardiometabolic health. So the SCFAs benefit us in two ways. The first one is they can help us directly maintain the intestinal epithelial barrier. And two, they indirectly regulate homeostasis. So this involves things like, as you see on the screen, regulating blood pressure, regulating lipid and glucose levels in the blood. And they're also involved in modulating the inflammatory and immune response. So they do these things via multiple mechanisms, which include modifying gene expression, acting as signaling hormones with G-protein coupled receptors, and so on. So via a combination of these systems, these SCFAs, as you can see, have a significant impact on various aspects of our basic functioning. The gut bacteria are also shaped heavily by lifestyle. So the three primary ones are diet, training, and drugs. So I'll go into these a little bit more. The first one is diet. So as you can see from this diagram here, this is talking about the impact of dietary protein, so plant protein versus animal protein on the gut bacteria. And as you can see here, plant protein consumption of primarily plant protein results in the upregulation or increased presence of bacteria which produce those very beneficial SCFAs, whereas high consumption of animal protein results in decreasing of those SCFAs. So this is because of fiber, especially plant protein naturally comes along with fiber, and this improves overall gut health, whereas animal protein doesn't have that fiber and it instead has more fat, which is linked to increased risk of cardiovascular disease and inflammatory conditions. Essential vitamin or mineral deficiencies or excesses can also cause gut microbial dysbiosis. And there's also increasing evidence that the use of preservatives in processed food can cause gut dysbiosis because they disrupt the mucosal layers of the gut intestinal epithelium. And so all of these changes are associated with increased inflammation and um, gut and the development of various health conditions. The second one that I mentioned is exercise or training. So regular exercise has been shown to have beneficial molecular adaptations that enhance cardiorespiratory fitness. Exercise also has positive effects on the gut microbiome diversity and metabolism and benefits for overall health. As you may all know, regular exercise improves cardiorespiratory fitness. So in particular, the exercise increases the prevalence of SCFA-producing bacteria from the Firmicutes phylum. And as I mentioned, these SCFA then promote anti-inflammatory activity. And then finally, drugs have a heavy impact on the gut microbiome. So these graphs here are describing the effects on the gut microbiome and various bacterial species after administration of antibiotics. Specifically, you can see this is D8 and then D180. So that's day 8 and then day 180 of an observation period after a 4-day antibiotic treatment. So as you can see clearly, long-term treatment with various antimicrobials can significantly diminish or alter the gut microbiome diversity, as you can already see this big difference in only 4 days of treatment. Antibiotic use can additionally promote the selection of antibiotic-resistant organisms and pathobions. As you can see, after the treatment, opportunistic pathogens increased in that second graph. Whereas, as you can see in the first graph, we had a decrease in many SCFA-producing bacteria, suggesting reduced anti-inflammatory activity. So a promising note, however, as you can see, is that it suggests that healthy gut microbiota are able to return to approximately pre-antibiotic state within 1.5 months. As you can see at D180 in the survivors graph, the bacterial species begin to creep back up and they're going back up, and the opportunist species begin to go back down again. However, you can see from that lost graph that there are several species that are unobserved, and this can remain for up to as long as 6 months. So this reduced microbial diversity is linked to a pro-inflammatory phenotype and disruption of the intestinal barrier, which increases individuals' risk of various diseases. So this really highlights the importance of proper antibiotic administration and proper usage by patients. So as I mentioned, disruptions of the gut microbiota have been implicated in a variety of diseases. These studies looking specifically at obesity, hypertension, and mental health issues. So this first one of obesity, this study looked at lean versus obese twins. And as you can see from this graph, the obese twins have a lower gut microbial diversity than their healthy counterparts. The same thing is noticed in hypertension. You can see that the hypertensive groups again have a lower gut microbial diversity than their healthy counterparts. This graph here, in the hypertensive group, I like that image, that graph there, because it actually shows which groups are enriched in the hypertensive versus the healthy control groups. And in the hypertensive groups, those bacteria, several of them are opportunistic pathogens, whereas in the control, which those are SCFA-producing bacteria, which would have anti-inflammatory activities. The same trend is even noticed in mental health conditions. So this study looked specifically at individuals with major depressive disorder. So exposure to stressors has been correlated with a pro-inflammatory gut microbial phenotype and an increase in systemic inflammation. This inflammation can impact functioning of the central nervous system, so indirectly causing issues like depression and anxiety. Additionally, the gut bacteria are able to modulate brain neurochemistry by directly modifying the secretion of various neurotransmitters like serotonin and adrenaline. So as you can see, the gut bacteria are able to very directly affect our health in a variety of ways. And a lot of research has been done on the gut bacterial states in disease conditions, but not so much in health. What we can see from these disease conditions is that there's two main trends, which is reduced gut microbial diversity and increased inflammation. So the same trends that I just mentioned are seen in aging. So as I mentioned previously, the gut microbiota is heavily influenced by lifestyle. but research increasingly shows that it is also heavily influenced by life stage. So microbial colonization of the gut actually begins at birth via the bacteria that are present in the mother's birth canal and on her skin. These communities will continue to grow and diversify until about 3 years of age when they start to stabilize into an adult type microbiome. And this will stay relatively stable throughout adulthood, excepting major diet changes or diseases, until this point in older adults, when the bacterial communities will begin to again change. And as I just mentioned, the same two trends are seen, which is increased inflammatory activity and decreased bacterial diversity. However, as I mentioned, the importance of lifestyle, research also has shown that good lifestyle behaviors like a healthy diet and regular physical activity can improve species diversity. So you see from this graph, these researchers looked at cardiorespiratory fitness, and as you can see, increasing fitness measured by VO2 peak is associated with increasing species richness. So this brought me to my research question, which was to examine and characterize the gut microbiota of healthy older adults in Saskatchewan, because as I mentioned, that there isn't a lot of research on the healthy state microbiome. So I want to explore what lifestyles are able to reduce some of the age-associated gut dysbiosis that has been shown in the literature. So we recruited healthy participants, and this is data that I'm presenting that was collected during my honors. So we started off with the demographics and medical history questionnaire that we asked participants to fill out online. And we had standardized questionnaires to assess lifestyle factors such as their mental health. So we looked at anxiety and depression and stress. We also looked at their sleep quality and their physical activity amounts and eating patterns and behaviors. Once participants had filled out the survey, we asked them to come in person to the lab to assess their cardiometabolic health. So we started off with cardiovascular health. We looked at their blood pressure. So this is an automated cuff that we put on their left arm. We did 5 measures and averaged the last 3. After blood pressure was done, we asked the participants to lie down in a supine position so that we could assess their arterial stiffness. This is measured via central pulse wave velocity. So as you can see, it's measured with these pen-like devices that are used to record the pulse at the carotid and femoral arteries simultaneously. And then we measure the distance between those points. And this is how we calculate the velocity of blood flow through the body. And as you can see from this helpful animation, a stiff artery is associated with a higher velocity, whereas an elastic artery has these contractions and expansions that are associated with a lower pulse rate velocity. We also assess participants' metabolic health via their, we looked at their fasting glucose and insulin via a fasted finger prick. We asked participants to be 12 hours fasted when they came in. So they did a finger prick to look at their fasting glucose. We also performed body composition assessments. We did 2 of them. So the first one is the bod pod, which is this egg-shaped device you see the person sitting in. And it uses air displacement to basically determine a person's body volume. And then it has age and sex-specific equations that it uses to calculate the person's body composition, so their fat and fat-free mass. We also use the C-cup picture here on the bottom. It uses bioelectrical impedance to again measure fat and fat-free mass and to determine total body water. At the end of the visit, we asked participants to fill out a 3-day food log so that we could get a look at their diet and see what they're consuming. So we asked them to fill out for 3 non-consecutive days to get an accurate representation of their overall diet. And then we used Chronometer to analyze their macro and micronutrient intake. We also asked participants to provide a fecal sample, which is what we use to look at their gut microbiota. So moving on to the results that I had during my honors. I'll start off with some participant demographics. So I collected 10 participants, 1 female and all of the rest are male. Their average age was about 61 years. Everyone was Caucasian, most were married, and all of them had at least a high school diploma. Several of them did have professional degrees because we had recruited heavily from the university. So on to their cardiovascular health. As you can see, all of their vital signs were good. Their systolic and diastolic blood pressure were well within the healthy values. Their pulse wave velocity was also good. The cutoff for older adults is about 10 for health, and most of these individuals were well below that cutoff. We also, as I mentioned, looked at the metabolic health. So waist circumference was all within the healthy range for both males and females. Their fat mass again was also within healthy range values. These values are actually these range values are actually taken for healthy young adults, and keeping in mind that these participants are 55 and older. So it was quite impressive that their health values, again, were within the healthy range for young adults. So this was what we had been looking for. So we were really happy to see this. Moving on to mental health, again, assessed via those questionnaires that I mentioned. So I'll first go over the structure of these graphs. The blue vertical bars are individual participant responses. The orange horizontal lines are the average of the participant responses, and then the green horizontal lines are the minimal cutoffs, so minimal stress, minimal anxiety, and minimal depression. And as you can see, almost all the participants were below even the minimal cutoffs for each of these for stress, anxiety, and depression. However, as you can see, we did have one participant who surpassed all of these values and did have quite high stress, anxiety, and depression. So for physical activity, this was based off of, again, another physical activity questionnaire. We asked participants to tell us how much physical activity they were doing. The recommended amount of daily physical activity for Canadian adults is 150 minutes of moderate to vigorous physical activity per week. As you can see, the majority of these participants exceeded that, in some cases by quite a lot. However, we did have 3 participants who did not meet those recommended guidelines of 150 minutes. On to their diet quality. This was a rapid eating assessment questionnaire to really look at the quality of diet that they were taking in, and processed foods versus whole foods, and so on. The maximum score is 39, and a higher score is associated with better diet quality. So as you can see, all of these participants had really good diet quality, again, suggesting healthy lifestyle behaviors. Now on to the fecal sample processing. So the fecal samples were processed via 16S ribosomal RNA sequencing. So we collect the sample from them, then we extract the genomic DNA, and then we amplify a specific region of the bacterial DNA using PCR. And then we, of course, size select to make sure that we're only getting bacterial DNA, and then we pull the samples together and we sequence them. And once we sequence them, we compare them to a database to identify which bacteria are present in the samples. So again, I'll first describe the structure of this graph because it will repeat a bit. So this is an ordination plot, and it's commonly used in sequencing data because there's so much data that it can be hard to visualize. So this is in 2D, and it's basically a rank-based system of how far apart different groups are from each other. So the further apart groups are, the less similar they are. And think of it like a Venn diagram, kind of, except with like multiple circles. So this one is of individual gut microbial differences, and each one is a different participant. So as you can see, they're not super similar. And this is kind of to be expected because people will have very different gut microbiomes, and they won't be exactly the same because they're heavily shaped, as I mentioned, by lifestyle and so on. So keeping in mind for the graphs going forward is that this is very much an exploration, hypothesis generating study. We wanted to explore what was there, and keeping in mind that this data is of a very small sample size, so I couldn't really do any correlation analyses. So the following graphs are all just preliminary data, things to keep in mind moving forward. So this is a graph looking at the gut microbial composition of people who used to smoke versus people who never smoked. And as you can see, it suggests that people who never smoked don't have very significantly different gut microbial differences than people who did smoke previously. However, keeping in mind that the people who did use to smoke had quit decades prior and had since been maintaining very healthy lifestyle behaviors. So, as I mentioned, a sign of a healthy gut microbiome is its ability to bounce back from changes and to come back to approximating normal. So this is not necessarily indicative of what might be the differences between people who are currently heavy smokers and people who are not. On to alcohol consumption. This is a graph of the gut microbial spread between people who do consume alcohol and people who don't. Again, keeping in mind that this graph is suggesting there are not significant differences. However, the people who did consume alcohol were not consuming significant amounts. They were consuming moderate amounts occasionally. So this is not necessarily indicative of differences in people who are heavy alcohol consumers. So, this is a graph looking at people in the differences of their physical activity amounts. As I mentioned, there were 3 people who didn't meet the recommended guidelines of 150 minutes of activity per week. So this graph seems to be initially going against what I said when I said that exercise has a significant impact on gut health. However, the thing to keep in mind with this is that these people were significantly active in other ways. They went out for hour to 2 hour long walks and so on every day. So this is suggesting that even any physical activity is beneficial in maintaining your gut health. It doesn't necessarily have to be very vigorous physical activity. And then moving on to the diet, this is when we start to see some more differences. So this graph is looking at fat intake, people who exceeded the acceptable daily micronutrient distribution range and people who didn't for fat. And there are some differences, but not as much. This requires looking further into what types of fat were they consuming and so on, and that might be the overlap between those two ranges, that people who were consuming more fat perhaps were consuming more of the healthier fats instead of the unhealthier ones. For carbohydrate intake, again, we had 3 people who didn't meet the carbohydrate intake requirements, and we see those differences, but it's not really a significant difference. Where it really highlights the importance of diet is here with the fiber intake graph. As I mentioned, the SCFAs, which are a vital source of very various functions in our body. So as you can see, people who didn't consume enough fiber are very, very different from people who did consume enough fiber. So I'll talk about that a little bit more here on this graph. So this is a community composition by phylum, which is the broadest level that you can characterize bacteria by. And as you can see in each individual, the most dominant phylum is Firmicutes, and this is a promising sign because research generally has shown that in older adults, you tend to have a decreased proportion of Firmicutes to Bacteroidetes. And so this is related with an increased prevalence of opportunistic pathogens from the Bacteroidetes phylum. So these people having a high proportion of Firmicutes is promising because it suggests that they don't have as many opportunistic pathogens. Firmicutes, they have high diversity in Firmicutes, which are the dominant SCFA producers. So again, suggesting that these individuals have less inflammation, overall healthier guts. This is supported by the results of their cardiometabolic values, like I mentioned, all of their vital signs, both cardiovascular and metabolic, were well within the healthy ranges, actually indicative of young, healthy adult ranges. So this is a promising sign. However, as I mentioned, there were some differences in health, for example, in the amounts of physical activity that people did and the mental health. Like I mentioned, there was that one person who did have quite high anxiety, depression, and so on. So moving forward, this is what I'm continuing on in my master's, is we are re-recruiting people, looking for a uniform cohort of 30 participants. We're adding a direct fitness measure. Instead of just asking participants how much physical activity they do, we're actually looking at their cardiorespiratory fitness via an exercise test. And the goal is to compare to North American databases of gut microbial sequences to see if really healthy cohorts are able to show that reduced gut dysbiosis compared to regular or normal older adults. Um, we also have a type 2 diabetes cohort in another study going on here that we will be comparing the the gut bacteria between, so we can really look for what are some of these differences in the gut microbial composition in a healthy state, old adult, and then in a person with type 2 diabetes in the same age. I'm thinking for future studies can also take more of a proteomics approach instead of just phylogenics to really explore the functionality differences in aging and health, because a big trend is that individuals will have different gut microbiota. They have different lifestyles, they consume different diets, they're not going to have the same gut bacteria. But often in a very healthy gut microbiome, what you have is um, overlapping in functions. So the important thing to look at in future studies would be to look at the functionality differences in aging cohorts versus younger adults, and then in aging healthy cohorts versus aging more, I guess, normal cohorts who don't do as much physical activity and so on. And I would like to thank you all for being here. Thanks to the funding sources, my supervisors, and all of our lab members.